So good morning, everyone. We are very sorry that we are not going to have Lucy in person here, but still, Greg and I met her in San Francisco this year, and her talk was so good that we were very positive that in our first workshop we would like to invite her to talk to us. And unfortunately, she even though she's not present, she sent us a video, a movie that she recorded a couple of months ago, talking about the subject that she was supposedly taught today. So just to let you know who is Lucy. So Lucy Collinson is head of electron microscopy facility at the Francis Crick Institute in London, UK. She has a degree and PhD in medical microbiology and carried out postdoc research at the MRC Laboratory for Molecular Cell Biology, UCL, and Imperial College London, investigating membrane trafficking pathways in lysosome-related organelles in mammalian cells, using light and electron microscopy as key techniques. And I'm sure that this is where her passion for microscopy initiated. She has run a series of biological electron microscopy facilities since 2004 at University College London, and then at the Cancer Research UK London Research Institute, which became part of the new Francis Crick Institute in 2015, where she is now, and where she oversees more than 100 research projects with more than 60 research groups within the Crick, imaging across scales from proteins to whole organisms. Her microscopy and technology development interests include volume EM, correlative imaging techniques, cryomicroscopy, X-ray microscopy, image analysis, and microscopy design and prototyping. She is also committed to open science through sharing of image data, protocols, software, and hardware designs. So now we are going to play Lucy's presentation. And if you have any questions, do not hesitate to put on chat, and we will send to Lucy, and we will respond later. Thank you.
Chris, can you give me a heads up? Are you seeing it?
Hi everybody, it's great to join you from London, and thank you very much for the invitation to speak today. I'm going to talk to you about our work on correlative light and electron microscopy, and especially moving into 3D using volume electron microscopy. So I run the electron microscopy science technology platform at the Francis Crick Institute in London, and we image samples across scales from cells to whole model organisms. But the thing that most of the samples have in common is that they contain something of interest, protein of interest, which is labelled with a fluorescent protein. And light microscopy is amazing because you can get dynamic molecular localization with green fluorescent proteins and its relatives, and you can watch things happen in vivo, like blood vessels growing and fusing, but you don't have any of the underlying context, so you can't see subcellular structures and organelles, and you effectively don't see anything that you haven't labelled. You're also limited in resolution, so even with new super...
Okay, sorry, wait 1 second, please. Let me try to figure out how to do this. Yeah, back here.
Resolution techniques, you can only see down to, I don't know, maybe a 50 nanometer, 100 nanometer resolution in vivo in cells and whole tissues. So then on the other side, with electron microscopy, your problem is that you're putting your sample into the vacuum of the electron microscope, so you can't look at it live. So it contains no dynamic information, and it's quite hard to label individual proteins in electron microscopy. But what you do have is really beautiful, highly detailed underlying structure. So you see all of the membranes in the cell, the organelles, even some large macromolecular complexes, depending on your sample preparation. And your resolution is typically in the range of 2 to 6 nanometers in the lateral direction. And then your depth resolution, the axial resolution, is given usually by the slice thickness of your sample, which is around 50 to 70 nanometers, so well in the super resolution regime. So the idea of correlative light and electron microscopy then is that you combine the benefits of both. So you take your fluorescent signal, which says where your protein is, and you combine it with your electron microscopy image, which shows you the morphology of the different organelles and cells. And that means that you can sometimes infer what your protein is doing. For example, if it's localized in a lysosome, then maybe it's involved in degradation pathways inside the cell. And when we're doing experiments, when we're working with our collaborators, we don't really mind what kind of light microscopy they use. And in fact, we recommend that they use the kind of fluorescence microscopy that they've optimized for their system. So if you're working on single cells, you might use wide field or confocal fluorescence. Or if you need very high resolution, you might use single molecule localization microscopy or structured illumination. But if you're working on whole zebrafish, or maybe C. elegans, then you might go towards light sheet. And then nowadays, we prepare the samples, get them into resin, which I'll talk about in a minute. And usually we'll put some kind of X-ray technique into the middle of our workflow. So these X-ray techniques allow us to see inside samples without destroying them. And so it helps us to identify our region of interest and to track it during sample preparation so that we can trim down to the much smaller field of view that we have in electron microscopy. And then the kind of electron microscope we use at the end to image the sample depends on the biological question. So if you're looking for something that needs very high resolution, maybe you need to see the 2 bilayers of a single membrane on a mitochondrion, then we would use transmission EM or electron tomography. Whereas if you really need to see your structure in 3D, whether it's a neuron in brain or a blood vessel in tissue, or even a whole endoplasmic reticulum in a cell, then we might use one of these new volume EM techniques, which incorporates block face techniques, serial block face SEM and focused ion beam SEM, and section imaging techniques with array tomography. So just focusing on the block face techniques, first of all, we have focused ion beam SEM. This is where you have a gallium ion beam, which is fired at the sample, so cells and tissues embedded in resin. And the gallium ion beam is inherently destructive and can mill away very thin slices of material. So we mill and then image the surface, and mill an image and mill an image. And in that way, as you can see on the right here, we build up a stack of images through the volume of the cell, in this case, a HeLa cell, which is at telophase, so it's dividing. Serial block face SEM is similar, except rather than cutting with a gallium ion beam, we cut with a diamond knife, which is held in a miniaturized ultramicrotome, and that's inside the chamber of the SEM. So again, we have a method of cutting here, the diamond knife, which moves across the sample surface, cuts a very thin section and throws it away. And then the SEM beam images the block face. So you cut with a diamond knife, image with the EM beam, repeat sequentially to build a stack of images through the volume. So the way that we use these volume EM techniques in correlative workflows is fairly tailored to the biological question. So this is an example of using it for imaging cells growing on cover slips, so that the sample is quite small. And here this is some work with Max Gutierrez at the Crick. So Max researches into tuberculosis, which is still a massive problem worldwide, especially considering antibiotic resistance. And the cell type here is cells, human lymphatic cells, which are primary cells, and they're infected with the primary pathogen. So everything is handled in biosafety level 3, which adds an extra level of complication to the experiments. So up here in the cell, you can see individual mycobacterium entering the cell because they're intracellular pathogens, and those are marked in red. So Max's group had some data. This is going way back to 2016. They had some data looking from right to left here to show that the bacteria, which are labelled in green, grow within these human cells. And over time, they divide and they're quite happy. But the peculiar thing was that at the same time, they're recruiting this protein called LC3, which is involved in autophagy. So we would expect that the recruitment of LC3 would lead to breakdown and recycling of anything that's inside that vacuole. So it's strange that the bacteria are able to quite happily grow and divide. So Max wanted to see what was happening at the ultrastructural level. So for single cells, they're plated into a dish. This is a Matek or an Abidi dish, and it's special because it contains a photo-etched grid of letters and numbers on the glass. And that means that after the fluorescence, live fluorescence imaging, you can effectively take a map of where your cell is in the sample. So here you can see it's in the grid square 9K, and we've marked the individual cell. And then you go through sample preparation for EM, which involves fixation with paraformaldehyde and glutaraldehyde, heavy metal staining with osmium, which also cross-links the lipids, dehydration with solvents like ethanol or acetone, and then infiltration with a liquid resin, which you polymerize at 60 degrees. And you can then, by dunking the block into liquid nitrogen, you can snap the cover slip off the surface and you get this beautiful imprint of the grid. And the black things that you might be able to just see at the surface are the monolayer of cells that we're interested in. So now we can use this grid map to find the same region on the block face, trim it down with a razor blade, and then mount it using some conductive epoxy glue onto a pin. And the sample then goes into, in this case, the serial block face SEM. So again, we have the electron beam imaging the sample surface, and then the knife taking a cut and discarding the section, and sequentially repeating this imaging and cutting process to build up a stack of images. So inside the microscope, that looks like this. We have the diamond knife here. The edge of the diamond knife is moving over the block face, which is here, and it's shaving off sections from the edge. This is the preparation phase. So we've actually placed the cell that we're interested in right in the middle of the block. And that means that we can trim towards the cell of interest, then close the door, set up the imaging run on a nearby cell, and we don't lose any of the cell of interest. And this is what the data looks like. So here we have the nucleus. The black is a layer of platinum on top of the block, and we're effectively going down from the surface where the cell is contacting the glass to the top of the cell. And you can see here these tubes are the mycobacteria sitting in this structure, and the shape of this quite nicely matches the fluorescence microscopy. So with these big structures that have a very unique shape, it's actually quite easy to identify them inside the cell. If you're looking to do some correlative, something that's maybe small and round like an endosome or a lysosome, this process gets much harder. So something that you'll have to do then is overlay the two data sets. Again, not so critical when the shape is recognizable, but extremely critical if you're trying to overlay something unknown. So what you do here is you take your fluorescence data and you drop in landmarks for regions that you can also recognize in your electron microscopy data. where you also drop in matching landmarks, and then you use a program like Big Data Viewer or Big Warp in Fiji or CLEM in ICI to bring those sets of landmarks into registration, which effectively warps the light microscopy data on top of the electron microscopy data, and you can do that either in 2D or 3D. The benefit of using these programs is that you can extract your landmark positions and you can unwarp your data, and you have a record of what you've done, which goes to reproducibility. So then we've got the two data sets on top of each other, so we can start to understand where the proteins are localized. And in this case, Matt in the lab did this process of manual segmentation to extract a 3D model from the stack of 2D slices. And this is extremely time consuming, even for what looks like a small volume of data. So here you can see that Matt has reconstructed the Mycobacterium in green, and then he's drawn around a membrane which was surrounding the bacteria, which here is shown in red. And that red then matches up to the fluorescence signal quite well for the LC3. But there's a problem, because in SEM we do not achieve the same resolution that we can get in transmission EM. And if you're now looking at the membrane level, you're not just trying to locate the bacteria, but you're interested in the limiting membrane that's containing those bacteria, then really you should be using transmission EM. So this is where we start combining microscopes into correlative workflows. So if we're effectively using our serial block face SEM as a smart trimming tool. So the block goes into the serial block face SEM, we cut an image until we can see our compartment of interest. This is now another sample containing the mycobacteria. And then the sample comes out and goes into a standard ultramicrotome where we cut ultra thin sections, pick them up onto the grid, and then image that in the transmission EM. So you can see the last image from serial block face here, and then the next section imaged using electron tomography. So now we have much higher resolution. You can see the nuclear envelope with nuclear pores, mitochondria. You can see the Mycobacterium tuberculosis sitting within now a single membrane. So that was unexpected, because autophagosomes were thought to have double membranes, which were characteristic of the compartment in electron microscopy, but more and more we're seeing these non-canonical LC3 labeled compartments that only have a single membrane. And so we've gone on and done some more work with Max's group, and Max has EM experts in his group as well now, who have continued to investigate this process of MTB compartmentalization and also escape of MTB from these membranes within cells. So that was looking at 3D correlative light and electron microscopy at the single cell level. But what do you do when somebody comes and asks you to find a tiny parasite in an entire model organism? So the problem gets much, much harder, because you have a huge volume to search through. And this problem we call finding the needle in the haystack. So I'm going to give you a recent example of some work which was done by Marie-Charlotte Demart in the EMSTP, and she was collaborating with Nagisa Yoshida, Serge Mostovi and Eva Frickel. And in this case, we're looking at toxoplasma parasites, so slight switch, but still looking at pathogens. And those are labeled in green here, and you can see them inside the zebrafish hindbrain. And then we also have a red fluorescent marker for macrophages. So the idea of the study then is to look at whether zebrafish can be used as a model system to study how the parasite behaves, and also the host-parasite interactions. So just a little bit of background. So toxoplasma is a parasite that infects, so its primary host is felines, and it's excreted by the felines and then taken up into intermediate hosts. So this is the reason why if you're pregnant, then you shouldn't clean out cat litter trays, because they may have consumed some toxoplasma and be excreting it. And it's there's thought to be a large proportion of the human population who are infected with toxo, but actually it only becomes a problem if you're immunosuppressed or in pregnancy. And just to say that Toxoplasma gondii is an intracellular parasite, so this is kind of similar to what we were looking at with Mycobacterium tuberculosis. So we're looking for the interaction of the parasite with the host cell. So the first steps here, now that we have a much larger sample, are to scan the zebrafish using micro-CT. So the micro-CT system is a little bit like a hospital CT system, but in hospital CTs, the patient lies on a bed and the CT system moves around the patient, taking multiple images, which are then reconstructed to give a virtual image of the patient. And that's usually to look at bones or maybe calcified plaques in blood vessels. With micro-CT, we actually embed our sample in resin, and they're heavy metal stained for electron microscopy, so the heavy metal stains give us really nice contrast. And on these systems, depending how large the sample is, we can achieve around about 1 micron resolution. So you can see the zebrafish hindbrain here with the eyes. So looking with micro-CT, we can find where our region of interest is, where the hindbrain is. But really, the Toxoplasma parasites are only a couple of microns, so they're really, you can just about see where they are, but it's really difficult to find them. So in this case, MC cut the hindbrain into semi-thin sections using a vibratome, and then imaged those individual sections by fluorescence microscopy. So now we have a limited volume where you can see the individual parasites in green and the macrophages in red. And she's also labelled the nuclei in blue, which helps to line up your fluorescence with your post-acquisition electron microscopy. So the image on the right here is now looking at the block face in the serial block face SEM, which contains this slice. And MC's looked for kind of gross morphological features like this line here, which you can see in both fluorescence and electron microscopy to align the two data sets. And that means she can take a good guess at where the parasites are going to arrive. So then she can locate those individual parasites. And doing that means that she can collect multiple images from multiple parasites or multiple needles in the haystack from within the fish. And what she finds actually is that many of the parasites are single. Some of them in coral here are replicating, which means that you can still see a connection between the two parasites that are sitting next to each other, and some of them are doublets, so they're completely divided. And the reason she can do that is because this is serial block face SEM, so for each parasite she has a complete 3D stack so that she can spot these areas of connectivity. So they went on and specifically targeted Toxoplasma parasites that were sitting within the red signal of a macrophage. And you can see from the serial block face SEM data set here, you have the kind of hallmarks of macrophages. So you have these big membranous compartments, which are formed from breakdown of things that the macrophages are digesting. And then inside, you can see two of these parasites sitting next to each other. So actually that tells us two things. So the zebrafish is mounting a response to the infection by uptake of the parasites into the macrophages. But this is also a doublet within a macrophage, so it suggests that the parasite can quite happily survive and divide. But if you start to get quantitative and move through the volume and look for other examples, then it turns out that most of these toxoplasma within macrophages, their membrane integrity looks pretty bad. So it looks like the macrophages are actually managing to break down the parasite. So there's lots of examples, and this is really nice to show that we're starting to move towards being quantitative in correlative light and electron microscopy. So the paper was submitted, and one of the referees came back and asked whether it was possible to see the host cell membrane surrounding the parasites. So whether the parasites had managed to break out of the parasitophorus vacuole, or whether they were being contained by the host. So what MC did was again use the serial block face SEM as a smart trimming tool. She cut down to the region of interest, identified parasites. We'll go through this one. So identified a parasite, two parasites that were sitting within a macrophage, and then moved the block from the serial block face SEM into the focused ion beam SEM, where we can get much higher resolution. So the Z thickness in serial block face SEM, or the cut thickness we use, is usually 50 nanometers, whereas in focused ion beam SEM, we use a cut thickness of 5 nanometers. And we image with 5 nanometer pixels, and that means that the data can be turned around in any direction because we have isotropic voxels, so 5 by 5 by 5 nanometers. So you can see the two toxoplasma here. You can see the really beautiful resolution on membranes. We have our fluorescence data here and matching XZ, XY and YZ planes on the left. And then this meant that the group could answer the reviewer's question. So in yellow, you can see arrows marking host cell mitochondria around the parasitophorus vacuole. And that's a hallmark of infection, which you also see in mice, which also suggests that zebrafish is a good model system here. But then the blue arrows are marking regions where this PV membrane, the host membrane, is missing from around the edge of the parasite, which suggests that the parasite is evading the host cell response. So I thought I'd spend just a little bit of time talking about data, because the techniques that I've shown you so far result in massive data sets, and that can be a real problem. So Martin, who runs the microscopy prototyping part of the EMSTP lab, has the rules of Big Data Club. So the first rule of Big Data Club is not that, it's actually this. So don't acquire big data if you don't have to, because it's really expensive. So it costs about, and this presentation, or this slide was from a couple of years ago, but it costs hundreds of thousands per petabyte for HPC storage. And something to bear in mind is that correlative imaging helps to minimize data collection. So you're effectively only collecting data in your region of interest. And we can take advantage of that in some of our tool development work that I'm not going to go into too much detail in today, but by being smart during our acquisition. So we have a research stream and development stream where we're putting fluorescence microscopes into serial block face SEMs, which means that we use a light microscope to automatically detect where our cells of interest are, and then we will automatically drive the serial block face SEM to only acquire EM images in those regions. So the second rule of Big Data Club is share your data. And there are ways of doing this now. So the EMBL-EBI, the European Bioinformatics Institute, have an electron microscopy public image archive called Empire, where you can deposit published data sets and also benchmark data sets. So if you go onto Empire and search for any of these, well, either my name, if you put in Collinson, or search for any of these numbers, then this will pull up a data set, which I didn't show you today, which looks at toxoplasma imaged by focused ion beam SEM from another paper, another collaboration with Moritz Treek's lab at the Crick. And you can download the data, and it also has associated metadata so that you can browse through either the raw data or our post-alignment data set and see if you agree with our conclusions. We have a really nice example of how sharing data can benefit both the people who collected it and other groups. So we had a collaboration with Oliver Flory at the Babraham Institute in Cambridge. And we did some correlative light and serial block face SEM to look at a process called entosis, which is cell-cell cannibalism, so cells engulfing other cells, which is a hallmark of cancer. So the data sets we collected for Oliver's project, he showed at a conference, and they were seen by Mike Overholzer, who spotted something in the data which was not Ollie's primary interest, but was useful for some work that Mike was doing on developmental cell clearance in C. elegans. So they effectively remined the data set and published it in a different aspect of it in a second paper. So the back end of Empire, where all of the images are stored, is now incorporated in something called the Bioimage Archive, which has launched and over the coming years will be built up to be able to take huge amounts of big image data related to the biosciences. And then the third rule of Big Data Club is to automate analysis and throw away the raw data. And that is fine in communities like particle physics and astronomy, where it's quite common. And there, the uninteresting data doesn't even make it to the analysis computer. But that's because they already have processing tools to extract their interesting features from the data set so that they can throw away the raw data. But we don't have the analysis tools to do that in biological electron microscopy yet. And the way we think you'd need to do that is, first of all, to collect ground truth data for machine learning to be able to automate analysis of the data. And then maybe you would be able to eventually throw away the raw data, although even then I'm a little bit skeptical because the data sets from volume EM are so rich that it's hard to know that you've mined everything of interest from the images. And we have a problem because collecting ground truth data here for us effectively means manual segmentation of the images. So we're going back to what I mentioned earlier with Matt drawing around individual Mycobacterium tuberculosis bacilli in serial images. So this is Anne in our lab, and she's drawing around the nuclear envelope of a HeLa cell in one image. And to reconstruct an entire nuclear envelope from a single cell would require her to draw around that structure in maybe 150, 200 images, which might take a couple of days if she was doing nothing else, but could take a week or more, especially for a non-expert. This process of manual segmentation is really important because unless we can see the data in 3D, then we can't necessarily interpret what it means for the biology. And these are just some of the papers where we've had to make models of volume EM data to interpret them. So here for vascular biology, for neurodegeneration, and for looking at the longevity of immune responses. But you think that understanding images is easy. So there was this famous study where the study leads managed to train some pigeons to spot pathological features in breast cancer images. So surely computers should be able to do it automatically. Well, in some cases, they can. So you can fairly easily extract the background, so the blank resin from around the cells in a pellet of HeLa cells imaged by serial block face SEM. So this is some work done by Martin in the lab. And you can see this beautiful reconstruction of the cell surface in 3D. But when we first started looking at this work, we were looking at serial images, serial transmission EM images collected of a dividing HeLa cell, and looking at these internal features of the cell, it's much more difficult. So we had a collaboration with a master's student at Imperial College who designed a specific algorithm for us that would recognize the nuclear envelope. And he did some beautiful work. So he noticed that the nucleoplasm has a different texture to the cytoplasm. So you can detect that using a variance filter. You can drop in seed points inside the nucleus and then flood fill to the nuclear envelope. And when it hits that barrier, then you drop in more seed points and flood fill, and then you prune back structures which look like they're attached in the image, but they actually aren't. So things like mitochondria. And you get a very nice overlay or nice segmentation of the nuclear envelope. But the problem is that as soon as you try and use that algorithm on an image collected on a different microscope, but from the same sample, then it fails. And the reason is that using a different microscope and different magnifications means that you have different characteristics within the image which can trip up the algorithm. So what we really need is machine learning and something that's generalizable across images collected from different data types. And this is a project done with another master's student initially who came from a computing degree, or sorry, a sandwich student who came from a computing degree at the University of Manchester. And it was inspired by advances from the neuroscience field. So neuroscience is where all of the volume electron microscopy imaging techniques come from, because they have the challenge of imaging neurons across large distances, but with the resolution to see individual synaptic vesicles. And likewise, they have a very defined image analysis problem, which is segmenting individual axons and dendrites through the volume of the data. So with a lot of labs focusing on a single segmentation problem and doing a lot of manual segmentation, including some citizen science games, they were able to collect a huge amount of ground truth data, which could be used then to train machine learning algorithms. The problem is that in cell biology, we don't have enough experts focused on a single problem. So we effectively want to be able to automatically segment all of the organelles in a cell, including the nuclear envelope, the nucleus, the nuclear envelope, mitochondria, lysosomes, endosomes, Golgi apparatus, etc., etc. And that's not even starting to consider specific organelles in specific cell types like melanosomes and melanocytes. So our approach is to collect reference data sets from different microscope types. So this is a HeLa cell pellet imaged by serial block face SEM, and we've imaged at 2 magnifications at 5 and 10 nanometer pixel size, because the images look different at different magnifications to computers. And those data sets are deposited in Empire, so you can download them and you can scroll through them and use them as you wish. And then, because we don't have enough experts to manually label each of the organelles in each of the images, we went to citizen science. And this comes from an effort to, well, I guess a similar problem where a field has a huge amount of data and not enough experts to manually label it. So in 2008, Chris Lintott at Oxford University set up Galaxy Zoo to feed out images of spiral galaxies to the general public. And he asked them to say whether there was a spiral galaxy present in the image, and then to say whether it was turning clockwise or anti-clockwise. And that project was phenomenally successful, and it led to a larger platform called the Zooniverse, which has millions of volunteer citizen scientists from the general public who work on a whole range of different projects across a whole range of disciplines, including astronomy, ecology, looking at historical documents, and now in the biosciences as well. So we have one of the first biomedical projects, and it's called EtchCell. So we feed out our serial block face SEM, focused ion beam and SEM images to the general public, and we ask them to segment organelles for us. And if you hold your phone up here, then it should take you through to our project. So this is a collaboration with Chris Lintott and Helen Spears, who's the biomedical lead and now a CZI imaging scientist at the Crick, and Martin Jones, who runs our microscopy prototyping facility. This is the interface. So we show our volunteers an image, we tell them what structures we're looking for, what microscopy is. We give them a flip book so they can go forward and back to a few images before and a few images afterwards. And they draw a line around the organelle. And it's a fantastic tool for public engagement. So it's gone out into schools, and here we have the Mayor of London drawing around a nuclear envelope for us at the launch of London Tech Week. But the million-dollar question is, can a non-expert segment as well as an expert, even to any level at all? Because if it's not the case, then it's no use to us. But it turns out that luckily we do see that at least some of our citizen scientists have some overlap with experts and can do as well. So this first project to segment the nuclear envelope went out to more than 6,000 volunteers, and we had more than 130,000 classifications. We feed each image to between 15 and 30 different citizen scientists, and you can see here all of the segmentations on a single image, and they're fairly varied in their quality. So one of the most complicated things for us was trying to take all of those different lines and work out which was the correct line. And it has some similarities to capturing underlying real paths from data from GPS. So this is trying to reconstruct the real layout of roads from GPS signals of cars driving along them. Unfortunately, those algorithms are proprietary, so we had to start from scratch. And the first thing we had to do was to remove graffiti. So we have some graffiti that I can show you. So this, we think, was somebody at the back of a school class. He wasn't really enjoying the activity, and some graffiti that we can't show you. So we have a first cleanup step. And then after that, we have an algorithm which was developed by our scientific computing core facility at the Crick, which then from the rest of the algorithm, from the rest of the annotations, finds a kind of average edge, which allows us to extract a single line. And it's very important to remind you here that we're segmenting the nuclear envelope and not the nucleus, which is an easier problem. So from that, we get a whole load of aggregated nuclear envelope models, which we can spin around in three-dimensional space. And then Harry, who was our student from Manchester, and Luke and Joost, who are in the scientific computing team, used that data to train the UNet. And what we see is the expert segmentation on the left, the aggregated citizen science in the middle, and the results from the UNet on the right. So already in 2D, you can see that they look remarkably similar. And then we have an addition where at the top and the bottom, it's difficult sometimes to tell what is nuclear envelope and what isn't, because it splits into small islands. And so we run the algorithm in 3 different directions over the data, and that fills in any gaps so that we end up with a really nice, clean, complete surface of the nuclear envelope. And then we can run that back on the original data, and some of those cells have been segmented by the citizen scientists and some of them haven't. The prediction step now takes less than 1 hour on this entire data set, which is pretty big. So it's 8K by 8K pixels per image and then 517 slices. So there's multiple cells in here. So for an expert to sit down and do this across the whole data set would have taken months. And this is the three-dimensional output from the machine learning algorithm. And the average house door distance, which gives us an idea of the accuracy, is less than 2 pixels or 20 nanometers from the experts, which is really amazing because even experts don't agree with each other on a pixel by pixel basis because of just the difficulty in drawing accurately with a brush on the screen. So just to say that the algorithm looks like it's generalizing really well. So this is also a HeLa cell, but collected with a different type of microscopy, and you can see that it's very cleanly picking out the nuclear envelope. And we're now running it on a huge range of different data sets that we've collected over the last years at the Crick, which includes tissue as well as cells, and making some tweaks. So all of this is available. The paper's now published in Traffic. It's Spears et al. It was out formally last week, although it's also on bioRxiv. And all of the data sets are in Empire, and you can find all of the segmentations and the code in GitHub. And just to say that we have plenty more for you to do on EtchACell beyond the nuclear envelope. So we also have Powerhouse Hunt, which is segmenting mitochondria. And the team recently entered Mito EM challenge for for mitochondrial segmentation from EM data and came 5th, which was very exciting because we're not specifically a machine learning lab, but our citizen scientists are doing a great job there. And we also have EtchACell VR for virtual reality, which is a collaboration with colleagues, including Rob Parton in Australia. And here we're taking the data from a single cell and reconstructing every organelle within that volume. And then our most recently launched project is EtchACell ER for the endoplasmic reticulum, which is a collaboration with Jez Carlton at the Crick. And so you can go online, and this is more difficult than the nuclear envelope and mitochondria, but you can go online and have a go at that project as well. So I guess the lesson, or what we've got to, is that you need different kinds of processors for different jobs. So you need to use the right tool for the right job, and you can parallelize specific tasks. So for some, parallel CPUs will be your best option. For some, parallel GPUs will be the best option. For some things, you may need to start with an expert, but that might not give you enough power. And so if you don't have enough power, you may well be able to parallelize that task as well by going out to the crowd and asking citizen scientists to help with your research. And Helen and Martin call that the HPU, or the Human Processing Unit. So with that, I'll wrap up. I'd like to thank you for listening. Again, if you want to have a go at any of our EtchACell projects and contribute, then you can either scan this or just search for EtchACell. I'd like to thank the guys down at the Zooniverse, and of course the entire EMSTP team. So I've specifically shown you some work today from Matt Russell and from Marie-Charlotte Demart. And from Martin and our physicists in the microscopy prototyping team, and also our Crick collaborators, and of course our funders.
Okay, everyone, Christina, okay, I think it was a very good, I really like her talks. I've heard her speak several times. It's like fantastic work they do there. Does anyone have any questions, or if you have questions, you can leave them in the chat and we can contact her. She said she's very willing to answer any questions. Kildare sent another message. He's very sorry that he couldn't participate today, but he's still not very well. But we will return this afternoon at 2 o'clock with Hosanna and Davidson. Okay. So thanks everyone for attending this morning, and we will hope we'll see you this afternoon. Thank you very much.